Biology 9700, paper 2, variant 2, May June 2024. Question number 1 to question number 3. And question number 1 is: smooth muscle is a tissue composed of smooth muscle cells. These cells contain cytoplasm packed with proteins that are involved in contraction and relaxation. Smooth muscles is present in the airways of the gas exchange system. Explain how smooth muscle cells in the wall of the bronchioles contribute to the function of these airways. And you know the bronchioles, at the end of the bronchioles, there are alveolus, and through which alveolus the exchange of gases takes place. And capillaries are around the alveolus, and the carbon dioxide from the capillaries enter into the alveolus, and the alveolus, the oxygen is entered into the capillaries. So in this way, the gas exchange takes place in the end of the bronchioles into the alveolus. So, and there is, you can say, the contribution of of these smooth muscles. They have, by contraction and relaxation, the gas exchange is possible. So they contract and relax for gas exchange. They control the diameter. So when they contract, the diameter is reduced. When they relax, the diameter is increased. So they also control the volume of the air, which is flowing in and out of these alveolus or the bronchioles. So in this way, these smooth muscles helps a lot by bringing out, by reducing diameter or by increasing diameter. Or secondly, they also control the movement of the air into and out of the alveolus. So in this way, they are very helpful. Next question, part B. And you can see, when viewed in longitudinal section, smooth muscle cells are elongated and taper at both ends. This is known as a fusiform shape. Each cell has a central nucleus, which also appears elongated. Diameter of a smooth muscle cell to show the fusiform shape. You can see the nucleus is shown, and it is 30 to 300 micrometer magnified. A student used a microscope fitted with a calibrated eyepiece graticule to estimate that the length of one smooth muscle cell was 250 micrometer. Name the type of microscope slide that is student used to calibrate the eyepiece graticule. And you know, eyepiece graticule is that scale which is kept into the eyepiece. So this is called as eyepiece graticule. The other scale, microscope or the microscope slide which is kept on the stage, is called as micrometer, stage micrometer. So both of these scales are calibrated, and they are used to measure the length or the diameter of any object which is placed on the microscope. So next question is, the smallest object this student can see without the use of microscope is 0.2 millimeter in length. Explain whether the student would be able to see a cell of length 250 micrometer. If we convert this 250 micrometer, then 250 micrometer can be divided 1 by 1000. So answer will be 0.25, which is larger than 0.2 millimeter. It means if we can see the objects which have having 0.2 millimeter, so the objects which are of 0.25 millimeter, they can also be seen. They are little bit larger than 0.2 millimeter. So the answer is yes. And next question, you can see figure 1.2 is a photomicrograph of smooth muscle tissue in the wall of the intestine. A capillary is visible in addition to smooth muscle cells. Yes, you can see the capillary is there. Uh, the this line of the red blood cells show the capillary, presence of capillary in these muscles. Outline the features that help to identify the blood vessel. And blood vessel, you can see it is of very thin walls. It is composed of very thin walls. So due to thin walls, you can see one cell thick, very close to tissues, and you can see the red blood cells are moving in a single row. So in this way, we can easily identify this is the blood capillary, in which is very only one cell thick, and it is very close to the tissues, and red blood cells are moving in one single layer, or you can say in the one row. Next question is, explain how the structure of a capillary is related to its function in smooth muscles. And you know, the function of capillary is one cell thick, is to exchange of gases. The function is exchange of gases. So its structure, you can say, is only, um, you can say, very thin walls, only one cell thick, due to which it is very easy for the gases to diffuse into the capillary or diffuse out from the capillary. So they have to cover the short distance due to, because only one cell thick. So they have to cover very short distance, and they have diameter is very small. Only red blood cells can pass, can move in single. The next question. Caldesmone exists in two different forms, H-caldesmone and L-caldesmone. H-caldesmone helps to regulate contraction and relaxation in smooth muscle cells, and L-caldesmone is found in some non-muscle cells where it also acts as a regulatory protein. Caldesmone is coded by a gene known as CALDI. CALDI has 17 exons decoding sequence. The primary structure of H. caldesmone has a repeating sequence in the middle of the amino acid chain that is not present in L. caldesmone. Researchers have discovered that a gene mutation is not the cause of the two different forms of caldesmone. So, explain what is meant by the gene mutation. Gene mutation is actually the sudden change sudden change in the sequence of base pairs in the DNA molecule. So the amino acid 1, amino acid 2, amino acid 3. So in this way, there will be the formation of a protein molecule. So sometimes what happens, the sequence of these amino acids is changed. So this amino acid another amino acid can be replaced this to number amino acid. So in this way, the sequence will be changed, and this is called as the gene mutation. Next question is there. Researchers now know that the two different forms of Feldesmon are the result of events occurring directly after transcription of DNA. Transcription of DNA changes occur to the primary transcript that is formed by DNA transcription, suggest how the smooth muscle cells and non-muscle cells can produce different forms of feldspar from the same primary transcript. Okay. Actually, this process takes place by the editing of RNA molecule, and due to this editing of the RNA molecule, the it can produce the different proteins from the same primary transcript in different cells. So alternative splicing creates multiple messenger RNA variants which translate into diverse proteins, and RNA editing alters RNA sequence to affect protein outcome. So in this way, it can happen, that is, we can add many, it can be added the different types of editing of RNA. Now I'm going to write the word I have told you. Editing in RNA. It can be brought about the primary transcription, or it can be done by the removal of introns, which are non-coding sequence, and by addition of exons, which are the coding sequence. So in this way, it can be edited. Suggest how the two different forms of Caldesmon can still have similar functions even though they have a different primary structure. Whether they have the different primary structure, but they are similar in having in tertiary structure, or the tertiary structure is 3D structure. So in this way, we can say that they have 3D structure, 3D structure, or tertiary structure, which is similar. So due to similarity in the structures, they have similar functions. Next question is, compare table 2.1 to name the cell structures that match the function stated. Do not use abbreviations. So manufactures ribosomal subunits from proteins and ribosome RNA. And you know, students, that ribosomal subunits are formed from the nucleolus, which is present in the nucleus. So nucleus synthesis triglycerides and other lipids. So these are the uh, smooth endoplasmic reticulum. And students, you know the difference between smooth endoplasmic reticulum and the rough endoplasmic reticulum. Rough endoplasmic reticulum have ribosomes attached on them, and the function of ribosomes is the protein synthesis. Whereas the smooth endoplasmic reticulum, they are not involved in protein synthesis. Instead, they are involved in the synthesis of triglycerides and other lipids. So they are having the different functions. Uh, Next cell structure is the organized microtubules of the cell cytoskeleton. So only these pair of centrioles which are performing this function. Now we are moving towards the next question. And next question is, figure 2.1 is a diagram of an alveolar macrophage macrophage. Some of the cell structure that would be visible using an electron microscope are newly formed phagocytic vacuole, phagosome, containing two cells of Mycobacterium tuberculosis, uh, phagocytic vacuole. You can see the phagocytic vacuole inside. Uh, the bacteria are there inside. The cell structure with the function are described in 2.1 is not shown in figure 2.1. Move back. Completing figure 2.1 by drawing and labeling the cell structure described in table 2.1. In table 2.1. This is the table 2.1. We have to complete the figure 2.1 by drawing and labeling the cell structure described in table 2.1. We have to draw these structures. That is, we have to draw the nucleolus, we have to draw the smooth endoplasmic reticulum, and we have to draw the hair in this way. The nucleolus can be formed. This is nucleolus. And second, which we have to make the rough endoplasmic reticulum. And we can draw in this way pair of centrioles. So it was all about the labeling and drawing. Now we are moving towards the next question. And next question is there. Tuberculosis can be prevented if the bacterial cells that have reached the alveoli are rapidly destroyed. So tuberculosis can be prevented if the bacterial cells that have reached the alveoli are rapidly destroyed. Alveolar macrophages can detect the presence of messenger M. tuberculosis in the alveolar space and can carry out phagocytosis to form phagocytic vacuole, such as the one shown in the figure 2.1. You can see the behind it was just like this. So outline the sequence of events that lead to the formation of phagocytic vacuole or phagosome, detection of the bacterial cells by an alveolar macrophage. So you can see how does it take place. What happens in this case, the during this process of the formation of phagocytic vacuole, the process of endocytosis takes place. And what is endocytosis? Endocytosis, the in which the macrophage cell surface, it surrounds the uh, bacterial cells, and in this way, the both sides of the cell membrane, they will join together, and there will be the formation of that side phagocytic vacuole. So in this way, they are formation of these phagocytic vacuoles takes place. So first of all, I would like to write the pathogens are the bacteria. Pathogen are the bacteria. And so what will happen around them, when they will enter into the body, so what will happen, the endocytosis, the process of endocytosis takes place when they will be detected on their detection. So this cell surface membrane, macrophage, cell surface membrane, membrane surround those bacteria and capture them inside. as you can see, uh, and there will be the formation of that. The membrane will move inside and it will catch them inside. So the process of endocytosis will take place. Now we are moving towards the next question. Name the cell structure that fuse with the phagocytic vacuole and release hydrolytic enzymes to destroy the bacterial cell. Yes, when they are captured by the cell surface membrane, phagocytic cell surface membrane, at that time, lysosomes are there which secretes the digestive enzymes or hydrolytic enzymes. They will break down those bacteria. They will destroy those bacteria uh, by secreting enzymes. So we are going to write the name of the lysosomes. These are the cell organelles which are producing these hydrolytic enzymes. Now, the next question is, research has shown that vaccination programs are cost effective and are very helpful in the prevention and control of TB. The programs may be aimed at particular groups of people that are at high risk of getting the disease, or they may be aimed at an entire population because the country has a high number of cases of TB. Or the Bacillus calmette urine BCG program or vaccine is a freeze-dried and contains live weakened Mycobacterium bovis. And you know, the vaccine contains the weak, or you can say live, weak, or dead microorganisms, which when they are a vaccine is when it is injected. So in the body, what happens? The antibodies are produced against them, and the memory cells formation of memory cells also takes place. So apart from being cost effective, suggest and explain the advantage of using the BCG vaccine for the prevention and control. And you know, in the body, what happens before when the vaccine is added into the body, injected into the body, at that time, there is the formation of antibodies, and memory cells are also present. So whenever those microorganisms enter into the body, so at that time already the memory cells are there. So there is you can say formation of the antibodies within no time. So in this way we can say they provide the active immunity, provide active immunities, and during these vaccinations, you can say uh, booster doses are not required during this process. And due to vaccination, we can the antibodies are produced immediately. Otherwise, you can say when any microorganism enter into the body, it will the body will take time, or the immune system will take time to detect those types of microorganisms which are new for the body. But when the vaccination is already injected, the microorganisms are not new for the body, so it will be very easy for the body to produce the antibodies against them. So it is very effective to neutralize or to kill the microorganisms which are entered into the body. So we can say this is very effective. Immune response will be against those microorganisms. Next, we are moving towards the next question. Question number 3. And the question number 3 is, lysosome is an antibacterial enzyme that was discovered in 1921 by Alexander Fleming, the scientist who discovered penicillin. Lysosome catalyzes the hydrolysis of glycosidic bonds present in peptidoglycan molecules to form smaller products NAG, N-acetyl glucoamine, and N-acetyl uramic acid. Before the induced pit hypothesis was produced in 1958, scientists believed that the lock and key hypothesis explained how lysosome catalyzes the hydrolysis of peptidoglycan to its products. Draw labeled and annotated diagrams in the space provided to show how the lock and key hypothesis was used to explain the mechanism of action of lysosome on peptidoglycan. Students, you know that according to the lock and key hypothesis, the enzyme always acts as lock and substrate act as key. Active sites are present. Active sites are present on enzyme on which the substrate attach itself, forming lock and key substrate enzyme substrate complex. So we can draw in this way, that is, we can draw an enzyme. This is an enzyme, and here can be the product. So enzyme, lock, enzyme, and this is substrate. They are complementary, they have complementary shapes. So what will happen? It will okay, in this way. So they will attach with one another and they will enzyme substrate complex. So at the end of the reaction, what will happen? The enzyme will it will remain behind, and there will be the two products, and one is NAM and the other product NAG. So in this way, enzyme substrate complex, they have complementary shapes, and as a result, when the substrate will join on the enzyme substrate complex will be formed, and as a result, at the end, there will be the products will be formed, which are NAM and NAG in this case, and enzyme will separate. It will remain behind. So in this way, this process takes place according to the lock and key hypothesis. The next question is, lysosomes and penicillin can be described as antibacterial agents. Both of they are the antibacterial agents. Lysosomes, they secrete the enzymes which hydrolyze the microorganisms, or you can say penicillin. which is an antibiotic. It will kill the microorganisms. So lysosomes and penicillin to show the similarities and differences between these two antibacterial agents. What of they attack on the bacteria, but in a different way. As you can see, we can say that the similarity is between them. Both of they cause the cell wall. They destroy the cell wall. Destroy the cell wall of bacteria by breaking bonds between them. And the differences, you can say that the difference between them is lysosome secretes enzymes which are protein in nature. And penicillin is an antibiotic which act as enzyme inhibitor. So penicillin prevents the formation of peptide bond. So in this way, they are similar and different from one another. It was all about from question number 1 to question number 3. Please like, share, and subscribe my channel. Thanks for watching.